Okay, well, we'll just move on then to our next two speakers who are going to talk about metals. First, we'll have Dr. Jennifer Nyland from Salisbury University, who will share some of her immunotoxicology and translational work with us. And then she'll just be immediately followed by Dr. Molly Kyle, who's from Oregon State University, who's leveraged data from the favorite data set of Dr. Davis and Haynes, as well as a cohort from Bangladesh. So thank you to you both, and we welcome you.
All right, well, thank you very much for this opportunity. I'm at a small comprehensive university on the eastern shore of Maryland now, and we don't very often get the chance to come over to this side of the big bridge. So thankfully the weather allowed that to still happen. So I am an immunotoxicologist, chemist initially by training, and so I have, don't worry, the slides are going to get a little bit lighter. An interesting and potentially different perspective on how metals, in particular, interact with the immune system and then with risks for infectious disease. And I'm going to try and tie it all into one mechanism, which is the inflammasome, and I'll explain what that is in a moment. So mercury has long been used as a component as an anti-infective agent. It's been used to treat infectious disease like syphilis and to combat infections. So I don't know how many of you might recognize mercurochrome. We had this in our house. It's a mercury-containing tincture that your parents would just paint on your skin when you fell and got scrapes, right? And it was because it was supposed to help prevent infection from setting in. And we also, this is calomel, was a certain number of pills per day of this fantastic mercury-containing compound would help prevent disease or treat disease. So there's a long history worldwide of using mercury-containing compounds to combat infection in the hopes that it would sort of boost your immune response and help you to survive these various interactions with your body, right? But those are inorganic forms of mercury. We're exposed to mercury in organic forms, methylmercury, through contaminated fish or rice consumption. But I'd like to tell you that all forms of mercury are immunotoxic, not just elemental mercury, Hg0, inorganic mercury, those mercuric chloride salts, methylmercury that you can get from contaminated fish and rice, and ethylmercury, that thimerosal that can be used in multi-use vaccines. They all affect the immune system. Now, they don't all affect the immune system in the same ways, and they don't all have the same level of toxicity. They're not all biotransformed and metabolized in your system in the same ways. But at some point, all of these forms of mercury can affect the immune response. And there is a vast amount now of cellular in vitro data, experimental in vivo animal model data, as well as epidemiological data demonstrating that fact. But the problem is that if you go through the literature and you cull it, there is really a lack of concordance or agreement in all of those different studies. We have a lot of evidence saying that mercury is neurotoxic. Lewis Carroll's Mad Hatter was mad for a reason. He was exposed to high levels of mercury. So the neurotoxicity side of things is very well established, but even at low dose exposures of mercury, there is neurotoxicity that can happen, and we believe that that's being driven by interactions with the immune response that's happening in the brain, those microglia or macrophage lineage cells. We also have a large body of evidence now demonstrating that interactions between mercury and some of these susceptibility or risk factors can lead to an increased autoimmune response. And we see that in some of the studies I'll show you in a minute, where individuals are co-exposed to mercury and some sort of immune trigger like an infection, and in this case, Coxsackie virus B3. We can also see that there is suppression of the primary immune response, similar to some of these other PFOS and PFOA that we just heard about, but that also happens with mercury, and that's evidenced in individuals who are exposed to mercury and infection for things like malaria. So these are different types of infections, different timing of infection, and potentially different species of mercury that are all interacting within animals, ecosystems, and humans in different ways. And those studies, you can't just take exactly what you're seeing at face value. You have to think about the range of exposures of the mercury, the co-exposures of what's also happening in that individual, whether it's a fish or a rat or a mouse or a human. And then, are there other infections, other drivers of the immune system out there? And so how is your immune system seeing that environmental exposure? What's the timing of it? So some of the first studies I did while I was at Hopkins, and this work began with Ellen Silbergeld and Noel Rose, so another incident of Ellen Silbergeld infecting our world. I did this fantastic schizophrenic postdoc with her, and Noel Rose looking at the effects of mercury on autoimmune disease that is triggered by a viral infection. And what we did in mouse models was we used a human strain of the Coxsackie virus and infected susceptible mice, so they have a particular genetic background that makes them susceptible to this particular infection. And they get a viral infection that looks just like the flu. They get all hunched and ruffled, and they don't want to leave their little nest. They want to stay nice and warm like we do when we have the flu. But over time, their immune systems are sufficient to clear that viral infection. And what happens when they have that viral infection is all these immune cells infiltrate into their heart, and the heart gets bigger, and it looks bad. But they clear the infection and those immune cells go away, and you would never know that those mice had any sort of viral infection or disease. But susceptible mice with a particular genetic background will go on to develop an autoimmune disease. And that autoimmune disease, there's no more virus, but their immune system, those cells move back into the heart, and they create this very big, flabby heart that can no longer pump appropriately. And eventually those mice will go on to end-stage dilated cardiomyopathy or heart failure. This same thing happens in susceptible humans who are exposed to this exact same virus. And in fact, this virus was isolated from someone who had this disease. Okay, so they go on, they get a viral infection. We get these infections a couple times a year. Most of us are lucky enough to be able to clear the infection and no disease. Some of us who have the susceptible background or perhaps some environmental exposures that make us at increased risks will go on to develop an autoimmune disease, and eventually we could need heart transplants. So we used this mouse model, and we said, well, what happens? We know what this virus model looks like, what this autoimmune disease that develops look like in particular mice. What happens if we add in mercury? But let's not just add in any mercury, let's add in a very low dose. We used what we felt was an environmentally relevant dose. We calculated it out. It was about a can of tuna fish a week for 2 weeks, a really low dose of mercury. And we gave that to the mice, and we tried at different time points. So we tried it just before we infected the mice, or we waited until they had already cleared the infection and then gave the mercury and looked to see what would happen. And what was interesting was that we saw a significant increase in the autoimmune disease, but not the viral disease when they were pre-exposed to mercury. So you had to have mercury on board first, infect with the virus. We would see no change early on in this acute phase of the disease. They would get the virus, but they would clear it. And it looked the same between those two groups. But then the ones that had seen the mercury, their disease was significantly worse for the autoimmune phase. They could not, their immune systems were over-exaggerated, over-exacerbated. So many, many more of them went on to develop that dilated cardiomyopathy endpoint. The majority of them had this inflamed heart from the autoimmune condition. And we were able to tie that to an increased infiltration of macrophages into the heart early on during the viral phase. So the immune response was different to the virus based on whether they had seen mercury or not. And we linked that to some pro-inflammatory cytokines, some of which are shown here. So this was an example of mercury, inorganic mercury, interacting with a viral trigger of disease to modulate the immune response and cause a very dramatic difference in the end clinical presentation of that disease. So in some epidemiological studies that I was fortunate to work with Renee Gardner, who is now at the Karolinska Institute, also in Ellen Silbergeld's lab, we were able to look at, so the interactions continue. We were able to look at, in humans, I called this the catch and release program. So we went out into the greater Hopkins community and asked mostly graduate students if they would be willing to let newly trained phlebotomists draw their blood. And we then took those peripheral immune cells, isolated them, and put them up in dishes, and then treated them with various forms of mercury at varying concentrations. And we repeated this many times. We stimulated those cells with this bacterial coat protein, the lipopolysaccharide, because we were trying to drive that macrophage response and see what would happen. And what we found was pretty interesting, that there was this, we called it an unopposed pro-inflammatory immune response. So the cells, isolated from these students, mostly people, when you put them with an increasing concentration range of mercury, so you can see that the top two panels are the inorganic mercury, the bottom two are the methyl or organic mercury, in increasing concentrations as you move along the x-axis. What I'd like you to look at is the slopes of these lines. So these are multiple individuals and comparing males to females, and we just looked at over a bunch of time, how could we predict what this response would be? How well could we predict that response? And what we found was that we would see an increasing slope for the pro-inflammatory cytokines as they were increasing mercury exposure, but that the anti-inflammatory or more protective cytokines being produced by our immune system were actually decreasing or flat. So it was an unopposed or uncorrected for pro-inflammatory immune response. And these are some of the same cytokines or small signaling molecules produced by your immune system that I was seeing in the viral studies in mice. So mercury on its own in this case was not able to induce this response. We had to stimulate these cells with that bacterial antigen initially. So these studies, what I'm showing you here is with LPS. When we don't have LPS in there, so no bacterial stimulation to the immune system, these low levels of mercury were not enough to cause a problem or to induce these cytokine responses. So similar to what we saw before, we needed both the mercury and the viral trigger. Now it's mercury and the bacterial trigger to induce these immune responses. So our studies were fairly consistent, but then when we moved on to the broader epidemiologic studies looking at populations in Amazonia and Brazil with some wonderful collaborations with researchers there, we were only able to find these same types of results in certain communities, usually those who were exposed to relatively high levels of mercury through small-scale artisanal gold mining. So why were we seeing these inconsistencies, not only in our studies as we moved from cell culture to animal studies and then to epidemiologic studies in humans, but moving into these the broader literature that's out there? If you really look at it, there are huge differences in exposure levels across all the different researchers and all the different exposed populations in the world. There's all kinds of different species of mercury that are used in studies or to which individuals are exposed. So we are not just exposed to methyl mercury through contaminated fish consumption, but also inorganic mercury, organic mercury in your dental amalgams, or in some of those fancy light bulbs that we have. So we are exposed to a lot of different species of mercury, and like I said, each of those can interact differently within your body, and so interact, and you can be exposed to different, your immune system can be exposed to those different species differently. But we're also, as has already been pointed out, a big gamish of all sorts of different things. We are co-exposed constantly to not just mercury and environmental toxicants, but other xenobiotics, other toxicants in the environment, medications that we're taking, various infections, and all of that underneath this big umbrella of who we are, whether that is our sex or gender, and other risk factors that we have, genetic risk factors. So mercury is interacting within that environment and modulating the immune response. but that mercury likely is insufficient on its own to induce these immune-related adverse outcomes. There needs to be some other trigger, and it could be many different things. So our work has recently moved towards trying to look more specifically at the mechanism of how it is that mercury is interacting with the immune response. So for a long time, it's been sort of this black box. Mercury is a metal. It's very simple, right? And so how is it that it's creating these very specific responses in the immune system in the context of these other exposures? And so some of our studies have started to focus on the inflammasome. So what's the inflammasome? Well, it's a multi-protein intracellular complex. These different pieces of the inflammasome can be floating around or induced within your immune cells, macrophages in particular, but lots of other cells. And its assembly is turned on and triggered through a variety of pathogen-associated or other cellular damage-associated mechanisms. That molecular machine that assembles inside those cells activates certain enzymes called caspases, which can then do different things. And one of the things that they can do is to cleave or cut apart some of the pro-cytokines and turn them into their active form. So this would be one of the initial stages to process within that cell a series of signals to allow for the activation, that unopposed pro-inflammatory immune response that I was talking about. So pathogen-associated, right, damage-associated, these are all signals that are coming from an infection or some of those other bacteria or viruses or things that are going on in our body, so those other triggers, right? How would mercury fit into that? Well, mercury likes to bind to sulfhydryl groups very tightly. So it could be that it's interacting with some of the proteins that are part of this complex and shifting how these individual proteins polymerize into this bigger machine. And so maybe it makes it work too fast, or it makes it work not well enough. There are, I have two very preliminary sets of data. I now work at an undergraduate institution, and so my researchers are all undergraduates, which means the pace slows, and occasionally you'll get some good data. So take this with a grain of salt, but I think it speaks to where we're going with things, and I think it's along the lines of some of the things we've seen in literature. So here we're using mouse-derived macrophages. It's a cell line, so take it with a grain of salt. It's a cell line. We're using a very short 3-hour exposure to mercury, in this case inorganic mercury, salts, and we're looking at relative cytokine expression levels, so RNA, so gene expression levels in these cells. And the order of these different bars is there's a control that has no mercury and no LPS signal. The next one has no mercury, but it's been given the bacterial signal. And the last one is, again, that low dose, about the same as 2 cans of tuna fish a week, mercury plus the LPS. And so, unfortunately, the student forgot to include the mercury, no LPS. Sorry. But so we have, what you can see is that there is a slight increase with the amount of interleukin-1, this pro-inflammatory cytokine that's being released or created by these cytokines, by these macrophages in response to the co-exposure of the bacterial trigger and the mercury. This looks very similar, but this actually is now looking at one of this, an inflammasome-related gene, so the NLRP3 RNA levels. And in this case, we saw a huge jump in the relative expression of that gene when we co-exposed to both the bacterial trigger and the mercury. So here's what's frustrating, is that other studies have demonstrated that the inflammasome activity is actually decreased with mercury exposure. Those studies, and our studies showed the exact opposite, right? So our preliminary data would show that the inflammasome activation is actually increased with that mercury exposure. The big difference is we're talking about a 1,000-fold difference in the mercury concentration. Theirs was significantly higher than ours. It doesn't mean you can't achieve that high mercury concentration in cells or in organisms, but we're trying to work at what we think is sort of what the majority of at least the US population might be exposed to. So using environmentally relevant doses of mercury, what we were showing is that there is an activation of this inflammasome. Our next step are to look at some of the proteins. These are genes, it's RNA expression, so what happens downstream, and are the proteins actually made at these differential levels? What are some of the regulatory mechanisms, and how is mercury impacting those? And then ideally what I'd really like to do is add in more of these co-exposures. So things like some of the polyunsaturated fatty acids that would come along with a methylmercury exposure through fish, as pertains to this meeting, viral exposures or bacterial exposures. And then one last thing I want to add in there is I'm always interested to think about, as Rod was talking about, where we are big bags mostly of bacteria. In your gut, if you have mercury methylating or demethylating bacteria at different levels, you could have very different responses and internal exposures to the mercury, and that can very much set up a difference in how your immune system responds to that exposure. The external dose would very much not equal the internal dose, and so would very much be different in terms of the immune response. So come over to Eastern Shore sometime and visit our gorgeous campus. Thank you very much.
I had one burning question for you, I'm sorry. Yeah, in your gene expression experiments, was that methylmercury or elemental mercury? Or what form did you use?
So in our gene expression, almost all of our studies, we've used inorganic mercury.
Okay,
so it's mercuric chloride salts, mostly because that tends to be the most immunotoxic. So we figured at our low doses, it would more closely approximate what would be happening in situ to tissues.